So let us discuss some interesting facts about the plasma membrane and its role in our day-to-day life. Plasma membrane is a special structure which covers the entire cytoplasm and cell organelles which are present in the cell. Now, cell organelles and the cytoplasm, nothing but the cytosol, entirely covered by the outermost membrane called plasma membrane or cell membrane. In other words, it is also known as sarcoplasm or sarcolemma. Sarcoplasm, which depicts cytoplasm, sarcolemma, which depicts the cell membrane or plasma membrane of the sarcomere, that is nothing but muscle fiber. Students, as we know that all the living organisms are made up of cells. Cells contains cell organelles, cytoplasm, and as I discussed with you, the cell covered by cell membrane that gives protection to the cell from external shocks and injuries. Now let us see uh, an interesting uh, an interesting concept with experiment. Before that, you have to know some interesting facts about plasma. Suppose cell required nutrients and water and also other materials for regular growth and development. Any nutrient or any liquid or any material should pass through membrane called plasma. Plasma membrane act as a barrier which prevents toxins or toxic materials without entering into the cell. Now, to understand the uses of plasma membrane, we need to know the solutions, different solutions which are required to the cell. Suppose take a saturated solution. Preparing a saturated solution. What is the use of saturated solution? In the experiment, I want you to explain. Take 100 ml of water in a beaker, then add sugar or salt. Stir it well until it is dissolved in the water. Now, if the salt or sugar dissolved in the 100 ml of water, then it is called as the saturated solution. Repeat it till little amount of it is left at the bottom of the beaker. Now, this is called as a saturated sugar solution. If you take the sugar or if you take the salt, then it is called as a saturated salt solution. Which one is more concentrated solution? Suppose, if you want to find out the concentration variation in between three different solutions which can pass us through the plasma membrane or cell membrane, let us find out. Take three different beakers with 100 ml quantity. Compare three solutions and answer the following questions. Now, in one of the beaker, we have to take a kind of solution, and in one beaker we can take the salt solution. In another beaker we can take sugar solution. In another beaker we can take another liquid. Now, half teaspoon of sugar to the first beaker, 1 teaspoon to the second, and 1.5 teaspoon to the third. So now for these three beakers which contains water, I have taken only the salt solution. Now to the first beaker, already I said that in these three beakers we need to take the water, isn't it? This is first one, second one, and third. So for the first beaker, half teaspoon of sugar we should add. To the second, we have to take if you suppose take a second 1 teaspoon of sugar we have to add. Whereas if you see the third one, to the third beaker we have to add 1.5 teaspoon of sugar. Stir these three beakers stir them well until the sugar solution dissolves. In these three beakers, which one is more concentrated? Let us find it. Easily we can say that the third beaker is more concentrated solution. The third beaker contains more concentrated solution than the remaining. Why? Because the quantity of sugar increases the concentration. Clear? The solution of which beaker will be most sugary? Absolutely, the third beaker. The third beaker, which contains more concentrated. Then, how can we make the solution of third beaker indicate to that of first one? How much water we should add to the solution in the third beaker to make it similar to the solution of the first beaker? Solutions with different amounts of solute dissolved in them are solutions in different concentrations. What I am trying to say, the solutions whatever present in these three beakers, they are having different concentration. The concentration is because of the sugar what we are adding in the water. Remember this point. Then after this, to understand the concept of concentration, let us do an experiment. Our aim is observation of materials in different solutions. So that is our experiment. We need some materials for this experiment. We need two beakers and a tap water, sugar, dry grapes, kismis, and these are the materials which we require. Procedure. Take 100 ml of water in a beaker. Keep the dry resin in it. If you keep dry resin or dry kismis in it, what will happen? Leave it for 1 hour. We have to wait until 1 hour and observe what has happened. Take it out and compare with the dry resin. Now, if you compare, have you find any difference between exact dry resin and the dry resin which kept in the water? No. You may try the same activity to slide dried carrots, other such vegetables. Did you ever observe that your mother dipped wilted vegetables in water? Why mother keeping the wilted vegetables in water? Have you ever seen while mother cooking curry, she was going to keep the vegetables inside the water? What is the reason? Can you identify the reason? Then take 100 ml of saturated solution of sugar in a beaker which was already prepared. Now, let us take another beaker. Already in the procedure I was mentioned. Let us take another beaker which contains the sugar solution. 100 ml of beaker we have to take. So in this 100 ml of beaker, what we have to do? We have to keep this resin. So this 100 ml of water contains the sugar. Concentration, I am not talking about concentration, but here I am taking a dry resin. So keep this dry resin inside this sugar solution. Let us observe what will happen. Now, here in the first case, in the first time, the swollen kismis of the previous activity of fresh grape or carrot, leave it overnight. So here what I have done, take the dry kismis, keep it in the sugar solution. Leave it for some time. After some time, what you can see or what you can observe. Let us find out